Just let me know if it is live.
Yes, it is live.
Okay.
No, but I need to check whether it is live or not. Okay. Yes, we are live.
Okay, then I will start recording now or after a few minutes.
Yes, you can start recording. No problem. Yes, we are live.
Should I begin? Or
just a minute, Shoma will ask you.
Yeah, I will ask you after the introduction. I will ask you and then you can begin.
Yes. So we should start. Okay, Priyanka? Okay. Welcome everyone to Bioingene webinar series. We are glad that it is our 50th webinar. We make it half century. When I start, I'm not thinking about that platform we can reach, but now we are confident that slowly we are reaching to more and more webinar. And thanks for everyone. Those are attached with Bioingene platform. I'm request all of you to make such a group that where we can discuss about plant science, agriculture, plant biotech, and everything. It's unique in that way that we can make a global community. We are very happy that today we have such eminent scientists in this platform. It makes really a good half-century webinar. So I request today Shoma to introduce our Bioingene little bit and speaker, and I will request Priyanka, and then later we go for the interactive session and question answer. All the participants, please make your webinar related questions, put your webinar related questions in the YouTube chat box. I will collect all the questions. And if you need any other information, you can email us at admin@bioingene.com. And after the question answer round, I will give a link for the feedback and certificate applications. Those are registered. Please put the registered email address. Mismatch of email address is very problematic for me to identify the actual participants. So I request Shoma, please start the Bioingene webinar.
Hello, everyone. I welcome all our viewers to Bioingene, a platform dedicated to the promotion of plant science research. The esteemed guest speakers on our platform are mostly plant scientists who present their research, and future scientists can gain knowledge, perspective, and inspiration from them. We do this through our webinar series, interview sessions, and publications. Thank you for being a part of today's webinar. It is special because we have reached 50 webinars. Today is our 50th webinar. As more people are joining in, let me provide some housekeeping information related to today's webinar. Please note that after attending today's talk, you can apply for a certificate of participation. For this, you need to submit the feedback form that will be provided multiple times in the YouTube chat after the presentation. When you fill out the feedback form, please use the same email address you used in the registration form, and remember to mention your full institute name and address. Mismatch in email ID may result in non-identification of participants, and your certificate may not be issued. You can collect your participation certificates after 2 to 3 days from our website. Bioingene does not send certificates through email. Please make sure that you have enabled YouTube chat on your device so that you can interact and submit your webinar-related questions. We will collect all the relevant questions for our speaker. Our esteemed guest speaker today is Professor Jonathan Lynch. Professor Lynch is a University Distinguished Professor at the Department of Plant Science, PennState College of Agricultural Sciences, USA. His area of expertise are crop ecophysiology, plant adaptation to abiotic stress, plant genetics, simulation modeling. His research interest focuses on understanding the basis of plant adaptation to drought and low soil fertility, including physiology, genetics, and ecology, centered on organismic processes. His current work focuses on understanding root phenes that can be used to breed crops with better acquisition of water, nitrogen, and phosphorus. He has published in many renowned journals and has received many awards and honors, such as the Non-Resident Fellow Samuel Roberts Nobel Foundation, the Distinguished Professor, Mexican Academy of Sciences in 2005, the Excellent Educational Work Prize from China in 1996, the China Friendship Award, the highest recognition awarded to foreigners by the government of China, the Fellow Crop Science Society of America 2004, Howard P. Taylor Lecturership in Root Biology Research in 2005, Alex and Jesse C. Black Award for Excellence in Agricultural Research in 2005, the Dundee Medal in 2015, the Woolhouse Lecturer Society for Experimental Biology in 2017, Dennis Hoagland Award, American Society of Plant Biologists 2018. He holds esteemed positions as the director, Feed the Future Innovation Lab for Climate Resilient Beans. He chairs the Root Biology University of Nottingham, and he is also a part of the UK Advisory Council Foundation for Food and Agriculture Research. We are highly honored to have him today on our platform. He will be speaking on the topic Roots of the Second Green Revolution. Professor Jonathan, kindly share your screen. Our viewers must be eager to hear you speak.
Thank you very much, Soma. I am sharing the screen. Can you see it?
Yes. Please start here.
Okay. Thank you very much for that kind introduction, Soma. Hello, everyone. I'm very honored to be with you today and talk with you about some of the potential solutions to the grand challenges you will face in your career. So the title of my presentation is Roots of the Second Green Revolution, Phenes and Phenotypes for Soil Resource Capture. So in the 1960s, if we fertilized wheat and rice, the principal global food crops, we actually lost yield because the plants got tall and fell over, lodging. Norm Borlaug and other breeders developed dwarf varieties of wheat and rice that could respond to fertilization without lodging and therefore have much higher yields with fertilizers. This was the historical basis of the Green Revolution. However, these innovative varieties did not yield better than existing varieties at low fertility levels. What I'd like to consider with you today is the possibility of developing genotypes of crops that have good yield at high fertility and better yield at low fertility. This green area represents the benefit we have already seen from the 20th century Green Revolution, and this blue area represents the potential benefit from a 21st century Green Revolution. Well, do we need a second Green Revolution? Here are some recent statistics. 770 million people suffer chronic hunger. 2.3 billion people suffer moderate or severe food insecurity. The undernourished population is expanding in Africa. Over 3 million children under the age of 5 die each year from hunger. In fact, half of all childhood deaths in the developing world result directly or indirectly from malnutrition. And of course, the human population is projected to reach 10 billion over the next 30 years. Virtually all of that increase will occur in regions that are currently food insecure. And in these regions, low input agriculture is a common occupation. In fact, it's the single largest human occupation on Earth. Low input agriculture is agriculture without the benefit of irrigation, pesticides, and fertilizers. And productivity in these systems is strongly limited by water and nutrients. So globally, the biggest challenge is drought. This is the current distribution of drought. Drought stress is expected to intensify this century as a result of climate change. Second global limitation is nitrogen. In this image, we see areas in green that have overloading of nitrogen. In other words, there's too much nitrogen being applied, resulting in environmental pollution. The areas in orange and yellow are areas that are in nitrogen deficit. In other words, the nitrogen is less from one year to the next. And in these areas, low nitrogen availability limits yield and crop productivity. And the third constraint globally is phosphorus. This image shows all the areas of the earth which have low soil phosphorus availability. In fact, this is a universal limitation for life on earth. Now, the first story I'm going to tell you is about this plant, common bean, Phaseolus vulgaris, which is the most important food legume globally, an important source of nutrients for poor farmers in Africa and Latin America. This shows bean growing in its native environment in the Andean region of South America. This is an eroded, volcanic, deforested landscape that is very low phosphorus availability. In fact, this plant you see on the right is severely phosphorus deficient. And our analysis shows this is the single greatest limitation to bean productivity in the tropics is low phosphorus availability. I began working on this myself in 1987. I graduated with my PhD and went to work at the headquarters for the International Center for Tropical Agriculture in Colombia, South America, where they had been working for over 20 years through brute force field screening to identify bean lines with better performance in low phosphorus soils than a Czech variety called Carioca, and they failed. So I was not satisfied with this result. I thought that breeding should rely on idiotypes and specific phenotypes rather than just yield in the field, because there are many things that affect yield in the field that are not related to phosphorus efficiency. So we began exploring mechanisms by which bean plants perform better in low phosphorus soils, with the idea that if we had an idiotype to guide breeding, that would be more efficient than this sort of brute force yield screening. So after many efforts, we decided it was the roots in beans that determine phosphorus efficiency. This is a traditional bean genotype. The roots are mainly growing down to get water. And now phosphorus is concentrated in the topsoil because it has very low mobility in soils. And so our idiotype had roots that were better at exploring the topsoil, more shallow roots. And these roots had root hairs with exudates. And as a result, they were better able to explore the phosphorus in the topsoil. So here we have the side-by-side comparison of good and bad genotypes and the topsoil foraging idiotype. Now, the topsoil foraging idiotype, as with many idiotypes, is composed of many distinct phenes. I'm going to be using the word phene in my presentation. Phene is an element of a phenotype. Phene is to phenotype as gene is to genotype. Phene is a word that is 100 years old, so it's almost as old as the word gene. So these are distinct phenes under distinct genetic control that all contribute to topsoil foraging. Using this idiotype to guide phenotypic selection with our partners in Africa and Latin America, we have been able to breed new bean lines with better root phenotypes for topsoil foraging that have over double the yield in low fertility soil in Africa and Latin America. For example, this is the release of 6 new varieties in Mozambique just a couple of years ago. This is our main partner, Magalhães Miguel in Mozambique, distributing the new bean seeds to Mozambican farmers. Compared with the best existing lines in Mozambique, these new lines had twice the yield, 3.5 times greater net income for the grower. They cut soil erosion in half because by having bigger plants, you protected the soil from rainfall. They had twice the nitrogen fixation. They benefit women, children, and men because each of these groups has distinct seed preferences. And they will be distributed in a region inhabited by 70 million people. We also replicated the success of the bean story in partnership with Xiaolongyan at South China Agricultural University. This is with soybean. Soybean is a food security crop in South China. South China does have low phosphorus soils, just as Africa does. You can see the image on the left. We have a traditional soybean variety that mainly has a taproot that goes down deep into the soil. And next to that, we see the topsoil foraging idiotype that has these shallow roots that are better for phosphorus capture. You can see here the current cultivar in the field in a low phosphorus soil in South China that yields much less than one of the new varieties developed by Xiaolong's team. So Xiaolong's team developed 15 new phosphorus efficient soybean lines that have been released in South China in regions inhabited by 600 million people. 3 of these phosphorus-efficient soybean lines were sent back to Mozambique, where they were being used there. So this illustrates the importance of root phenotypes and the possibility of using root phenotypes to breed better plants for low phosphorus soils. The next story I want to tell you is about this plant, maize. Maize is also an important source of nutrition for low input farmers in Africa and Latin America. This is summarizing some years of our publications. On the first column, you see the phene that we're focusing on, and then the yield gains we see related to variation in these phenotypes under drought, nitrogen stress, and phosphorus stress as a percent. So you can see some of these are very high. So in other words, root cortical parenchyma, as I'll be showing you today, is associated with between 80 and 800% increase in yield under drought. So some of these numbers are very significant. And I won't review all of this today. This is all published research. I'm just going to be highlighting a few case studies. So nitrogen leaches downward into the soil, as you can see in this simulation. The red color here is nitrate. This is environmental conditions in Pennsylvania in the USA. And in the spring, the rain is washing that nitrate down deeper into the soil as the roots are growing. And the risk is that that nitrate will wash out the bottom of the rooting zone before the plants can acquire it. And so our idiotype for nitrogen and also water capture is different than that for phosphorus. Phosphorus is shallow, whereas nitrogen and water tend to be deep. Our idiotype is therefore steep, cheap, and deep. And I'll be explaining that in a moment. Steep refers to architectural phenes. Cheap refers to anatomical phenes that affect the cost of growing roots, and deep is where we want the roots to be. Let me give an example. And here you see on the image, computer simulation of a standard maize root phenotype on the right, and on the left, the steep, cheap, and deep phenotype. Let me give an example here of the steep component by fewer nodal roots. What do I mean by this? These are maize roots grown in the field in South Africa. And you can see there's a difference in the number of these axial roots. These are called nodal roots below ground. Nodal roots are called crown roots in this species. And you can see in this case on the left, there's few, and on the right, there's many. In fact, if we evaluate 5,000 different genotypes in the field, we see large variation for this phenotype between 5 crown roots per plant and 35 crown roots per plant. So we take this to our computer simulation model, OpenSimRoot, to see if this is worth exploring in further detail. Here we have simulations of a maize root system on the left with 6 nodal roots, and on the right with 36. And this is in the range of variation we find in nature. And the simulation told us that actually the one on the right, the one with many nodal roots, was making too many roots that were competing with each other for carbohydrate inside the plant and for nitrate outside the plant. So this is overproduction of roots. You can see here, if we zoom out, and you can see this from more distance, 46 nodal roots, they actually are less developed and less deep. If we only have 6 nodal roots, each root is able to acquire more resources and have more carbohydrate, is able to grow better, and you end up with overall a more vigorous and deeper root system with better nitrate uptake. This is what the computer model told us. Of course, this is a guide to our research, but we do not stop with simulation. We take it to the field for confirmation. So here we have reduced crown root phenotype and its effect on yield under drought. Now, Pennsylvania is a region where precipitation is common. So to create reliable drought conditions here, we use these automated field rainout shelters. They have a rain sensor that pulls the plastic cover over the field when it is raining. And when it is not raining, it leaves the field, so the field is an open air. And in this environment, we grew maize lines, natural variation for crown root number going from, in this case, about 25 to almost 45 per plant. And in agreement with the computer simulation, we found that the lines with fewer crown roots had significantly deeper rooting, better capture of deep water, which we determined with O18 labeling, better leaf relative water content, much better leaf photosynthesis, greater biomass, and 57% greater yield. We did the same kind of study under nitrogen stress, in this case in a field in South Africa. You can see the high nitrogen plots in the background that are green and large, low nitrogen plots in the foreground that are chlorotic and smaller. Once again, natural variation in crown root number was associated with deeper rooting, better capture of deep nitrogen as we determined from N15 labels, better nitrogen accumulation, leaf and canopy photosynthesis, better growth, and 180% more yield. So indeed, fewer crown roots is good for water capture and nitrogen capture. However, remember, phosphorus is a topsoil resource. It's in the shallow part of the soil. So in that case, we do not want deep root systems. And in this case, we find the opposite. We see a trade-off. We see for phosphorus capture, you want more crown roots. So here's the same experiment done in a low phosphorus field. And in this case, you see that the lines with more crown roots had better phosphorus acquisition, better biomass, and 120% more yield. So this is one of the reasons there are such diversity for architectural phenotypes in plants, is that there's trade-offs for contrasting resources in time and space. Let me give you an example of what I mean by cheap in this idiotype. Cheap is reduced root respiration. And one example of this is root cortical parenchyma. This is not a computer simulation model I am showing you. This is laser ablation tomography, which I'll talk about in a minute. So this is a maize root grown in the field, and we've ablated it with lasers, and we can see this green area is root cortical parenchyma. It's airspace, and there's significant genetic variation for this phenotype. In fact, these two maize lines are recombinant inbred lines that descend from the same two parents, so they're sisters of each other. In this case, this genotype has an intact cortex. In this case, this genotype has converted many of these cortical parenchyma cells into airspace, parenchyma. And our thinking was that by converting living cells to airspace, we make the root much less expensive in terms of carbon cost, nitrogen cost, phosphorus cost. So we begin this analysis in our in silico computer model. So here's open SimRoot. This shows root cortical parenchyma, RCA, with the warmer colors being more RCA. It's developmentally regulated. The model knows how this phenotype affects respiration, nutrient content, nutrient transport, and so on. And the model tells us that RCA should benefit nutrient capture. So here we have a simulation in a low phosphorus soil of identical phenotypes differing only in RCA formation, root cortical parenchyma formation. Then you can see over time, the line with RCA ends up being larger because every unit of root is a little bit less costly in terms of carbon and phosphorus. So it's able to grow further into the soil, acquire more phosphorus, which feeds back on greater photosynthesis. There is a canopy photosynthesis model as part of open SimRoot, although it's not visualized. And so a small difference in efficiency of each root segment results in a large difference over time of soil exploration. This is what the simulation told us. Once again, we take this to the field. Here's our field rainout shelters under drought. And we found that these lines with less RCA, these are all recombinant inbred lines of each other, so they're closely genetically related. Lines with more RCA under water stress, and the lines with more RCA had cheaper roots, had much deeper roots, better water content, better growth, and 800% more yield under drought. You can see these low parenchyma lines are very poor under drought. These ones look pretty good with the same amount of water. Now, this was a study in experiment station conditions, of course, with simulated drought. We wanted to confirm this under more realistic conditions. Joseph Chimunga was recently able to do that. This is on-farm studies in Malawi where we went to farms where women were growing maize and then worked with the women farmers to phenotype the roots. And then we returned at the end of the season to determine what was the yield of those lines. So this was a study of maize lines grown by women farmers in their own farms. There's very little scientific intervention. And this was, we looked at 10 lines in 14 farmers' fields in each of 2 regions of Malawi where there's natural drought. And what we found was that the high RCA lines had better water content, better biomass, and between 78 and 143% more yield under drought. Under nitrogen stress, you can see here again is our field in South Africa, high and low nitrogen, natural variation in RCA. We see that the lines with more parenchyma had cheaper roots, deeper roots, better nitrogen content, better photosynthesis, better growth, and 58% more yield. Now remember in the case of phosphorus, I showed a trade-off in that phosphorus was shallow, water and nitrogen were deep, and so there was a trade-off in root strategies. In the case of rhizoeconomic phenes like Arenchyma that make roots cheaper, there's no such trade-off. And so here we see phosphorus. This is work of Tania Galindo Castañeda. So here we have the high RCA lines, low RCA lines in this low phosphorus field. You can see the high RCA lines did better. And you can see here the yield gains. So the lines with more Arenchyma had less respiration, deeper roots, better phosphorus content, better biomass, and 75% greater yield. So I've shown you that root cortical Arenchyma is capable of reducing root respiration. That's shown here. This is root respiration. So Arenchyma can cut root respiration in half. This is rooting depth. It significantly increases rooting depth. And this is yield under drought. It doubles yield under drought by having deeper roots. Once we realized that cheap roots were useful, we thought of other phenes that could reduce the cost of soil exploration. For example, cortical cell file number, which is simply the number of cells, parenchyma cells produced in the root cortex. You can see there's natural variation for this. And this on the left is fewer cell files, on the right more cell files. And once again, by having fewer cell files, cheaper roots, we cut respiration in half, we significantly increase rooting depth, and we double yield under drought. A third phene we published about is cell size. The idea being that large cells are mainly vacuole, and so they're cheaper to maintain than cytoplasmic tissue. And so here we have natural variation for cell size. Maize lines with larger cells had half as much root respiration, deeper roots, and double yield under drought. So whatever we do to make roots cheaper apparently makes them go deeper and therefore improves performance under drought. There have been thousands of papers studied about water use efficiency, in other words, how shoots use water to gain carbon. And what I'm showing you today is we perhaps should also think about carbon use efficiency, which is how roots use carbon to gain water. Now, everything I've shown you so far are things that we've already published. I want to show you some new unpublished results. So we continue to discover new phenes that may be interesting. In this case, parenchyma cell wall thickness. This is the work of Jagdeep Singh Sidhu. And you can see Jagdeep found significant genotypic variation in maize for the thickness of the cell walls. You can see here on the left a genotype with thick cell walls, and this one has thin cell walls. So we're using a different simulation model to evaluate this. This is called root slice, and this allows us to evaluate structure function relationships at the tissue and subcellular scale. And you can see here's root slice simulations of a maize line with thin parenchyma or cell walls. Here's 2 microns thickness versus 4 microns thickness, and that's well within the range we observe in nature. So root slice predicts that thicker cell walls will reduce cytoplasmic volume. So as we increase cell wall thickness, the cell wall volume in a section of root increases by 65%, but the cytoplasm decreases by 36%. Because the cytoplasm decreases, there should be a cheaper, metabolically cheaper tissue. So the number of mitochondria in that root segment should decline by 35%. The amount of phosphorus in that root segment should decline by 53%, and the amount of nitrogen should decline by 54%. That's what the simulation tells us. So we evaluate this in real plants. So these are real roots here that we're measuring with varying cell wall thickness. And we see the thicker the cell walls, the lower the respiration, whether we measure this per gram or per length. And so these are significant reductions, 34% less respiration per gram, 23% less respiration per length. And indeed, when we take these lines to a natural drought environment, we see that lines with thicker cell walls do significantly better growth under water stress. You can see here we're going from about 48 grams of biomass to 75 grams of biomass associated with cell wall thickness. So this should be an interesting phene for breeding. It's also under genetic control. This shows you that we found a locus on chromosome 3 of maize associated with this phenotype, and the allelic variation in this locus is associated with cell wall thickness, so this is a promising genetic lead for molecular breeding. I want to mention a second new phene, which is multiseriate cortical sclerenchyma, which was recently published. So this is what a normal maize root looks like. And if we zoom in on the outer cortex, you see this is what the normal parenchyma tissue looks like. And Hannah Schneider discovered that some lines look like this, where the outer region of the cortex actually has these very thick cell walls and small diameters. And this is actually sclerenchyma, sclerified tissue, that's embedded with lignin. You can see here lines with MCS. MCS is multiseriate cortical sclerenchyma, have much more lignification than lines without MCS in that particular region. And because of this lignin concentration, you see the greater lignin concentration, the greater tensile strength these roots have. They're stiffer. You can see here the tip bending force, the force required to bend the tips is twice as great for MCS lines than non-MCS lines. So these MCS lines have more lignin, the roots are stiffer, and they're able to penetrate hard soil layers. So here we have lines without MCS, those with MCS, we put them, expose them to a wax layer, and we have twice as much penetration of roots through the wax layer in the MCS lines. And in the field, where we have compacted conditions in the field, we see under non-compacted conditions, the MCS has no effect on rooting depth or biomass, but under compacted conditions, the MCS lines have significantly greater rooting depth and significantly greater biomass. I should point out that Hannah found this phenotype in multiple cereals, including wheat, in addition to maize. MCS is genetically controlled. You can see here there's some loci from GWAS. So this is a trait that's interesting candidate for breeding. Interestingly, Yvonne Lopez Valdivia discovered that in maize roots that were 5,280 years old, found in a cave in Mexico. Now, this is in the early days of maize domestication, so we don't know what these shoots look like. But already, now wild maize, teosinte, does not have MCS. Wild wheat ancestors do not have MCS. So MCS seems to be something brought in by human selection. And you can see already 5,000 years ago, this maize root had, here the lignification shown in red. Here we have this MCS tissue very clearly. So already early in domestication, MCS was selected for, which suggests that it has some utility. Perhaps it's an adaptation to human managed soils. The third new phene I want to mention of interest for breeding is Xylem perforation plates. So this is maize. So here we have a longisection showing a Xylem vessel, and here's the length of the Xylem vessel. Here's the perforation plate that borders two adjacent Xylem vessels. Here's a radial cross-section, and you can see here what the perforation plate looks like. It's a little bit of a constriction of the Xylem lumen. Chris Strock discovered phenotypic variation for this in maize. So here we have a short metaxylem vessel versus a long metaxylem vessel. And the short metaxylem vessel was associated with a tall perforation plate, whereas the long metaxylem vessel was associated with a short perforation plate. So we wondered if this would affect xylem function, and indeed, hydrodynamic modeling suggests it could affect function. So here we're modeling xylem vessels with tall perforation plates, and you can see from the color spectrum here, these tall plates significantly reduce the flow of water through the xylem vessel, whereas the short perforation plates do not restrict the flow of water through the xylem vessel. To confirm the modeling results, Chris measured the theoretical maximum axial conductance from the Hagen-Poiseuille equation by looking at cross sections of maize roots. And then he actually measured the axial conductance of these roots by applying a known pressure head. And he compared the two, and he found that the longer the vessels were, the less difference there was between the actual and potential conductance. In other words, the longer vessels were actually more conductive in nature. Now, taking this to our rainout shelters, here we see an aerial view of the 5 rainout shelters, and we see that there's no effect on yield of this phenotype when the plants are well watered, but under water stress, we can more than double yield under drought by having these long vessels. So here's another phene that may be useful for breeding. Now, when I'm presenting these sorts of results, I don't want to give the impression that these phenes act in isolation, because the fitness of an organism is determined by its integrated phenotype. I give the example of your hand. Fingers are very useful in terms of your ability to function as a human. Thumbs are very useful, but if you have thumbs and fingers together, we can do great things and build rocket ships and do science and stuff like that. So that shows you the example of phene synergism, that the utility of a phene is a function of the other phenes in that phenotype. And as an example of that, I want to talk about the first case study, the synergy of metaxylem anatomy and root architecture in Phaseolus. Now here's Phaseolus vulgaris that we've talked about already. This is common bean, most important food legume on Earth. This is a close relative of common bean, Phaseolus acutifolius, which is cultivated by people in the desert southwest of North America. And being a desert adapted plant, it has a deeper root system. So here's what the root phenotypes look like. Here's common bean and here's tepary bean, which goes deeper. Now in both species, there's phenotypic variation for the conductance of these roots. So here we see a root cross section with relatively few metaxylem vessels, and here's one that is full of metaxylem vessels. So both species have this variation. So Chris was looking at the potential synergies between the anatomy of xylem and the architecture of the roots. So we grew these in a desert environment in Arizona, and here you can see tepary bean acutifolius, and you can see the acutifolius just by these images grows better when the plants have high conductance, xylem, versus low conductance, whereas the opposite is true of common bean. Here you can see in these images of common bean, the common bean grew better under drought when it had low conductance xylem versus high conductance, and keeping in mind that the water is more available with depth. So here's the actual data. So in tepary bean, as we decrease the xylem vessel, we have greater drought stress. In other words, greater xylem conductance was beneficial. Whereas in the shallow rooted species, common bean, greater xylem conductance was harmful. So very different effect of xylem conductance depending on the root architecture, which is a totally distinct phene under distinct genetic control. This illustrates that you have to think about integrated phenotypes in your breeding. Let me give you another example of that, and that's phene synergism drives nitrogen efficient root phenotypes in rice. This is work of Ishan Ajmera. Ishan developed a rice implementation of open stem root. It's very impressive considering rice is such a complex root system. So Ishan identified, this is IR64, a Green Revolution rice variety. That's a very impressive rice cultivar released by Erie. And he identified these other rice root phenotypes that should give us better performance of low nitrogen soils compared to IR64. So over the past 30 some years and over the next 80 years of projected climates, Ishan looked at the yield of these phenotypes compared to IR64. IR64 here is shown as this purple line. And you can see a very significant gain in yield, up to 80% gain in yield in future climates compared to IR64 for performance under low nitrogen. Now, one of the nice things about in silico studies is you're able to dissect your results and understand exactly how you got the results you did, because you have full access to information, unlike with real plants. And so here we're looking at phene synergism, which is greater than additive effects. So once again, instead of thumb being 1 and fingers being 1 and thumb and fingers just being 1 + 1 = 2, maybe it's thumb 1 is finger 1, but having thumb and finger together is 10 in terms of fitness. So it's a synergistic benefit. So here we see shoot dry weight for these lines. This is IR64 in purple, and the actual shoot dry weight was better than IR64 for these different clusters. However, if we look at simply their expected response, just by adding up the individual contributions of the phene states, they're worse than IR64. In 2 cases, they equal IR64. In the other cases, they're worse than IR64. IR64 is a great variety. It's hard to beat IR64. The reason these lines were better than IR64 is because of synergism. This is the synergistic benefit. So we had significant synergism among these phene states, and that accounted for the benefits rather than an additive effect. These results and others show that the utility of a phene state is influenced by interactions with other phene states in an integrated phenotype. So for breeding for disease resistance, we can just add together the resistance alleles from different sources and have a resistant plant. That's not true in this case. We have to think about how the organism functions as an integrated phenotype. And this is a challenging problem. For example, if each of the 5 phenes of interest exist in only 4 states, in other words, very steep, intermediate, shallow as an example, well, there would exist 4 to the 5th or 1,024 integrated phenotypes, each of which should be evaluated in different environments. So this is a problem that's too large for empirical research. So to help us address this problem, we've been using in silico approaches. I want to show you an example, multi-objective evolutionary optimization to explore phene space using OpenSimRoot. This is work of Harini Rangarajan. And so this is multi-objective evolutionary optimization. So Harini looked at how maize and bean phenotypes would perform across a range of nitrate and phosphorus availabilities. She looked at 22 root phenes, each in 5 states, which results in a decision space of 5 to the 22nd power, evaluated under 25 levels of nitrogen and phosphorus. That equals 6 times 10 to the 16th phenotypes. So obviously well beyond the capabilities of empirical research. She was optimizing against 6 objectives. She performed 250,000 evaluations to finding 2,000 optimal solutions, and this required millions of hours of computational time. So one of the conclusions from Harini's work was that phenotypes balance trade-offs among objectives. So here we see, for example, 4 maize root phenotypes, and here's how they perform against the 6 objectives she defined. Here's the 6 objectives here. So for example, this phenotype did very well for biomass and pretty well for these other ones, but did do pretty poorly in terms of root length at depth. Whereas this one over here has very strong in terms of root respiration, but then not so well in terms of phosphorus uptake, et cetera. And so what we see is there's no one ideal phenotype that does everything well. That's why nature is so diverse. And so this is another conclusion from this type of analysis. There are multiple co-optimal phenotypes for each objective. So there's more than one phenotypic solution to a complex set of constraints. And once again, this explains the diversity we see in nature. And this must be acknowledged in our research and in our breeding programs. So I want to go now to one of the key bottlenecks for deploying root phenotypes in breeding, and that is the challenge of root phenotyping in the field. So first I'll talk about root architecture in the field, shovelomics. And so basically this is simply using a shovel, excavating the root crown, rinsing it, and then imaging it. So here's a bean root crown excavated in the field. This takes about 2 minutes per plot per genotype. In a normal year, our team will do this for 25,000 plots per year for a variety of different species. This is not difficult. Working in collaboration with Alex Bush, Alex developed DIRT, which is digital imaging of root traits, which allows you to take an image of these phenotypes, upload them to a website that Alex has created, and then it will return to the user an array of architectural metrics. And a number of people are using Alex's tool. Let me show you an example of how this works. So here we have variation for growth angle of crown roots among maize lines. So here we have a more shallow crown root, and here we have a steeper crown root. And here again, when we look at 5,000 different maize genotypes in the field, we see very large variation from maybe 20 degrees from horizontal to 90 degrees. So very shallow to very, very steep. So Hannah Schneider led this team effort over many years. So 4 years of phenotyping, 480 maize genotypes in South Africa, measuring the angle, then conducting a GWAS, genome-wide association study. We identify a locus on chromosome 1 associated with this phenotype. This locus codes for a gene that we know is expressed in roots. And now a mutant in this gene, which is CYPK15, is actually steeper. You can see the wild type versus the mutant here. This is the root angle as measured under low nitrogen and drought conditions. Under both conditions, the mutant is steeper. And that because of its being steeper, it should have better nitrogen capture and growth under low nitrogen. Open SIMRU tells us that the change in angle should result in better nitrogen uptake from the soil and better biomass under low nitrogen conditions. When we grow these plants in the field under low nitrogen conditions, we see in terms of nitrogen 15 in the shoot, which is a measure of deep nitrogen, we applied a tracer. Under low nitrogen conditions, we had significantly better nitrogen capture by the mutant, which resulted in better biomass by the mutant. And so this is an example of how we can use fairly crude field phenotyping of root architecture to find genes. I want to now talk about root anatomy in the field, anatomics. So this employs laser ablation tomography, which we invented here in my lab at Penn State. Basically, it uses a high-powered laser to ablate tissue. This is a real-time image of a maize root that is being ablated. So it's being destroyed by the laser, but then we have an image of everything that was in that tissue. And so we can do all kinds of anatomical analysis. We subject those images then to image analysis and RootScan, which is a free software developed by my lab. RootScan allows you to quantify things like parenchyma here in red or Xylem here in yellow and other things. So here again is an example of gene discovery from field phenotyping using this platform led by Hannah Schneider. So we have maize roots excavated in the field. We ablate them on the laser shown here. We subject them to image analysis in RootScan. Then we take this data to conduct a genome-wide association study. We find a locus here in chromosome 2. We know that that is expressed in the root cortex where it should be for parenchyma. Then developing overexpression or RNA interference lines, we see that RCA is affected by altering this allele. A CRISPR line has much, in this gene, has much less parenchyma formation, and a mu line, a mutant line in this gene has much less parenchyma function. So here again, an example of a gene discovered BHLH121 by means of this field anatomical phenotyping platform. Finally, I want to mention LEADER as a way to measure root depth in the field. Here's Molly Hanlon, who led this work, holding a X-ray fluorescence spectrometer. Now, the X-ray fluorescence device is able to detect metals in objects. And so holding it up to a plant leaf, you can measure all these metals in that leaf. And the idea is that deep-rooted lines versus shallow-rooted lines will have differences in leaf element accumulation. So LEADER stands for leaf element accumulation from deep roots. So let's say that the blue element is more in shallow soils. The red element is greater in soil depth. And so what you'll find using this handheld device is that both shallow and deep lines will have the blue element, but only the deep line will have the red element. Now, using this method, we can also apply a tracer in the field. We've been using strontium. And then if the line has in the leaves has tracer, that means that it's reached that level in the soil. So the idea is a non-destructive determination of root depth through this leaf analysis. Now, we've conducted field tests across the United States to see how this works in a range of maize lines. You can see some of the environments here in different soils in the US. And what Molly's found is the actual rooting depth measured with soil coring here shown on the x-axis, estimated rooting depth shown on the y-axis, and she sees a very good correlation between the actual and estimated of r-squared 0.76, which is very nice for field work. And this method is able to discriminate deep and shallow rooted lines through this XRF spectra. So in summary, natural genetic variation for root phenotypes presents excellent opportunities to develop crops tolerant to water and nutrient stress. Topsoil foraging enhances phosphorus capture, while subsoil foraging enhances nitrogen and water acquisition in leaching environments. Several anatomical and architectural themes affect the metabolic cost of soil exploration, which is an important component of soil foraging efficiency. Bean and soybean genotypes incorporating root phenes for superior phosphorus acquisition are being deployed in Africa, Asia, and Latin America. The steep, cheap, and deep idiotype for water and nitrogen acquisition in maize is promising. New phenes are being discovered that improve tolerance to abiotic stress. Laboratory and field phenotyping platforms exist for important root phenotypes. But coming back to the second green revolution, breeding for yield at low fertility has been successful in the few cases it has been attempted. Aluminum tolerance is being deployed in breeding programs. Phosphorus efficiency is more complex, but as I showed today, it's working. Nitrogen is quite complex, but it's promising. Manganese tolerance is working. I'm not aware of any breeding programs that are selecting for potassium, calcium, magnesium efficiency, but they should be feasible. Recent advances in root biology are making it feasible for the first time to design specific root systems for specific target environments. The bottleneck at this stage is phonetics, i.e. understanding the phenome, rather than genetics, in my opinion. We need more information about long-term system impacts of this technology, but it should be benign. For example, there's no resistance breakdown. If we develop an insect-resistant or disease-resistant genotype, over time, the insect or disease agent will evolve a mechanism to overcome that resistance. That does not happen with abiotic stress. A drought-tolerant line will always be drought-tolerant. It's not scale positive, meaning it does not just benefit rich farmers who have access to inputs, unlike the first green revolution. And there's an incentive to reduce pollution, environmental pollution in rich countries. This is important since rich countries fund most of research in plant biology. As soils degrade, fertilizer becomes costlier and the world population expands. The need for this will only grow in years to come. Now, the work I'm showing you is obviously the result of many, many people. This is the current configuration of people in my team. Thank you very much.
Thank you. We have collected few questions for you, but after the interactive sessions, we'll discuss on that. But most of people are asking, and also my point of view, that what setup you need in your lab, like your pictures, your anatomical figures are so informative. By looking, you can identify each and everything of a root. So many people are asking in that respect, that setup of your lab. I'm sure you covered in that your presentations. It's few is a ledger-based technology, your computer simulations model, and obviously microscopic things. So because we are amazed in use your slides of sections of root. So definitely it is a common question to everyone. And another question is changing in lignin or changing in the modification in the structure in root. Is it somehow deal with the penetration power? If you hollow roots make the less penetrative or not? And another is changing in the structure, it can reduce the water or capillary actions of water intake in the plant. So these are three related questions. You can go, we can collect more and go after your presentations.
Okay.
After you interact.
Thank you, Shubha. So should I respond now? Is that what you're asking?
Yes, if you willing to give the answer.
So the first question I think is about how we do root imaging. Is that right?
Yes.
And so, you know, we have always just done what we felt like we had to do, right? So I didn't begin my career thinking of myself as a root biologist. We started looking at phosphorus in bean, and we looked at mycorrhizas and root exudates and shoot processes, and it seemed to us the most important thing was roots, so we began looking at roots. And then the challenge was, well, how do you see roots? Because they're either growing in an artificial environment that's not realistic, or they're growing in soil where you cannot see them. That's why we developed shovelomics, so we can just dig them up and look at them. And then we had the question of, well, now I'm seeing differences, but what does it mean? So what? I mean, so we see variation. How does this affect performance? So we developed OpenSimRoot so we could ask those kinds of questions. So, and then when we realized anatomy was important, we thought, okay, how can we easily measure anatomy? Because in those days, everything was hand sections, and it took many, many hours
Yes.
to do a genetic studies. I mean, we had people 8 hours a day who were just sectioning. It was like a meditation practice or something. I don't know how they did it. And we had the idea, oh, we could use a laser to do this faster. So basically, we did not sort of think about all these things in advance. We just had a problem. And so then we came up with a solution to that problem. So now we can, consider roots in soil through open simroot and root slice. We can measure them in the field through shovelomics. We can measure the anatomy with laser ablation tomography. So we have a set of tools. Leader was another one. It's very difficult to measure root depth. Coring is very laborious and very noisy, very messy. Nobody wants to do it. So that's why we developed Leader. So we just basically have had to confront challenges, and the solutions did not exist in the literature and the community. There are not very many people looking at these issues, or if they're looking at them, they're looking at them in what I consider to be very unrealistic ways, like, in little pots in the greenhouse or something. And so we just sort of came up with our own solutions, and I'm sure, many of the people listening will come up with even better solutions in the future. So that was the first question, right?
Yes. Second was the structural modification of root can hamper the water absorptions and penetrations.
Absolutely. And that's, basically, when you start looking at roots, you're confronted with a huge phenotypic variation. You look at a maize plant, they all look mostly the same. There's not too much variation above ground. Below ground, there's variation for everything, right? And so when we start looking at anatomy, we see, for example, in the case of MCS, many lines have no MCS at all, and others do have it. So here's a phenotype that modifies lignin, stiffness, penetration. They either exist or does not exist. So huge variation for that. Likewise with Xylem, I showed you several pictures in this presentation in bean and in maize. Large differences in Xylem that affect performance depending upon anatomy and other things. And, surprisingly, I mean, this is for the young people who are there. I mean, you've spent your whole life listening to professors like me or reading books or papers, and this is sort of received knowledge. This is the body of knowledge we have. But you have to realize it's very incomplete, right? I mean, there are many things we don't know. And maybe the important science is things we don't know, rather than things we do know. So there was dogma about, Xylem vessels and conductance, and yet we see that, well, there's actually variation for length of Xylem vessels and the perforation plates and the amount. So all of this variation is the tools that's providing tools for us to breed better plants, right? And I think we really need crops that are totally different than the ones we have today to meet the needs of 10 billion people in a degraded global environment. And young people, the future plant scientists, really have to have new ways of thinking. Of course, yes, molecular biology is important. I'm sure you agree with that, Shuvo. Yes, genes are important. But I think at this point, we have wonderful tools for molecular analysis, genetic analysis. The thing we lack really is sort of the imagination and the ability to think about what phenotype we want. Maybe the maize of the future is only a meter high and it has reflective trichomes and it associates with nitrogen fixing organisms in the rhizosphere. Maybe it's perennial. I mean, maybe it's very different than the maize we have today. And so, yes, the molecular genetic tools are in place, but do we know, what kind of plant we want? I think that's where I see a big opportunity.
Sir, another question just came up that root common beans, they asking that root, is it behave similarly at early stage and in older stage? So it's age of the root. I think it's a answer regarding the age of roots during the aging, that how much changes are occurred.
Well, this is another complexity in root studies. Roots are so much more complex than leaves in many ways. And one of them is that every leaf on a maize plant is more or less the same, although some might be more in the sun and some might be more in the shade. If you look at the anatomy of the leaf or so on, it's all about the same. But roots, especially in dicot plants like bean, change dramatically with time. The single root axis changes dramatically. In the monocots, we have root cortical parenchyma formation, root cortical senescence, other changes that happen in that axis. So it no longer requires water and nutrients. It's mainly just a conduit connected to the mother plant. And in dicots, you have secondary growth. I haven't talked about that today, but we published some papers about that. That's the thickening process that woody plants have. And as the thickening occurs, the cortex disappears. And then you just have a woody, xylem, phloem conduit with no ability to absorb water and nutrients. And so, yes, they change dramatically. And that is something that's built into our simulation models. It's built into our idiotypes. Is how can we, how do we want this to work? Do we want a plant with more or less of these age-related processes? Another variable is, think about a maize plant. Once again, we've researched this, but I haven't talked about it today in the interest of time. It starts producing roots that are very small, the seminal roots, and then each node produces more roots that are thicker and thicker and thicker. So you see the brace roots coming out of a maize plant above ground are as big as my finger. So there's large variation among root classes, you see. And in fact, Hannah's work on CYPK15, the gene that affects root angle, it only affects the angle of 2 nodal positions, not the others. And so you see, it's really quite complex. And when we think about even the genetics, we say, well, it's not that all roots get steeper. No, just a couple of positions, but that is enough to affect function. So we can think about an idiotype, instead of thinking all roots are steeper, all roots are more shallow, we can say, well, these ones are steeper, but these ones are even more shallow. We can design our own architecture if we want to. So there's many, it's a high dimensional decision space. That's the way the computer people talk about it.
And there is no questions related to directly. I'll look on to that questions in that stage. So I request Priyanka, please go for the interactive session, and so we can collect more questions and listen the journey of Jonathan. Thanks.
So this is a great pleasure and honor to have a 50th question and answer round with Professor Lynch. So without further delay, I would begin with the first question, my favorite one. We would love to know how you built your career. Could you please share your journey as a plant scientist with our viewers?
Okay. When I was 9 years old, I saw on the news that there was a famine in Africa, in Biafra, in Nigeria. And I thought this was terrible. And I had the idea that this was related to bad soils and bad food production. So I determined that when I grew up, I was going to do something to make better plants for these difficult soils. That's when I was 9 years old. And that's what I did. As an undergraduate, I studied soils and plant nutrition. And in graduate school, I studied plant nutrition, and my job at CIAT was in plant nutrition, and my position at PennState is in plant nutrition. So my whole career has been oriented towards finding ways to, develop crops for these difficult environments. So that's a very short story, I guess, Priyanka. But
yes. So what motivates you to work for the betterment of agriculture?
well, I think it's natural for, to be sympathetic to people who are suffering and to want to do something about that. You see a huge number of people have food insecurity and poverty, and it's going to get worse in the future. So if we're able to do something, and of course, I was fortunate in being born in a rich nation where there's resources where we can do this kind of research. Some nations really don't have the capability. The very poor nations cannot conduct this type of research. It's very expensive, or they don't have strong universities programs. So I'm in a position where I can help people. So it's been very meaningful for me to work with many colleagues in Africa, Asia, Latin America to have these benefits and to train them, because I've had over 100 students I've trained, and they are now throughout the world having tremendous impact, more impact than I have had. So this is perhaps the most rewarding thing about this type of work, is working with young people who can then go and have their own careers and have their own impact.
So you mentioned that you begin as a like plant nutrition, like your research begin with the plant nutrition and then you move to root studies. So according to you, which sector of plant science research will prosper in the near future?
Well, I think really the grand challenge for humanity in the 21st century is how to sustain 10 billion people in a degraded environment. And we all know the environment is getting worse because of global change and soil erosion also, and deforestation and all these things. And we have 10 billion people. You know, how are we going to do that? We cannot do what we have done in the past. That will not work. Already it's not working for all these hungry, poor people, right? And it's only going to get worse. So I think in this case, we as plant scientists are in a unique position to make a contribution because our civilization depends on plants, on food, fiber, and fuel from plants. The major human interaction with the Earth's biosphere is cultivating plants in soil. That's what we do. That affects the atmosphere. If we're going to bring CO2 out of the atmosphere, plants, right? If we're going to feed ourselves in these future environments, plants. So we're really at the juncture of, of what humanity needs to do in this century. And there's so much that needs to be done that has not been done. Like I was saying a moment ago, young people, you may see all this information in the textbooks in your classes and you think, oh, everything is known. No, I mean, there's many things we do not know. Something as simple as what is a drought tolerant plant? I mean, that's a very complicated thing. We don't really understand. And maybe a drought tolerant maize plant is different than a drought tolerant rice plant. They're different organisms, right? So there's so much to be done. And so plants and the interface of plants and the environment, I think, are really central. And specifically, the idea of stress is the idea of most plants on Earth, even in rich nations like the US. Our yields of maize are not even half of yield potential. In Africa, they're only 5% of yield potential. So, it's not that photosynthesis itself is the problem or something like that. It's that the plants are under stress. And that's the issue is how do we help plants deal with stress, with not enough resources, with too much heat, not enough minerals, not enough water. I've always felt like that was the challenge, especially when you go to developing countries, but even in rich nations. Like we saw in the nitrogen map, India, for example, uses a lot of nitrogen. China uses a lot of nitrogen, too much, really. So in that case, okay, nitrogen limitation is not so much an issue, but excessive nitrogen issue. So if we understand how to make plants more efficient, in the US, the estimates are that only less than half of the nitrogen applied as fertilizer is ever taken up by the roots. The rest is just causing pollution. So if we make a more efficient plant in India and China, we can use less fertilizer. That's less environmental impact. So I think this issue of, plants, plant biology, agricultural plant biology, and getting plants to resist stress, I believe that is a very fundamental challenge for humanity in this century. And it's a difficult challenge for young people. It really is a difficult challenge. On the other hand, I think it's a wonderful opportunity for young people, the people who are listening, because you can have such a huge impact with your work, right? There's so much that can be done. So if you could make any contribution to this problem, you will benefit many, many people in the present and also in the future.
So the next question you already answered quite a bit. It's what are your thoughts on climate change and global warming effects on agriculture?
Yes, well, it's going to get much worse because the major economies are really not addressing it aggressively enough. And so it's a problem right now, and it's affecting people right now, and it's going to get worse. Even if humanity decided today to become very serious about this, because the lag time involved, there will continue to be problems of heat and drought and all these associated things for the future. So it is a very serious challenge. I believe it is a more serious challenge than most people understand. If you look at the literature by the scientists rather than what the government say, some of the scenarios are quite alarming. In fact, I showed you just briefly today an image of drought stress where it is today on Earth and then drought stress in the future. And if you spend time looking at that image, it is terrifying. There's no Amazon forest. There's no rain-fed agriculture in the US and in Europe. how does humanity survive under these conditions? I mean, this, like I'm saying, this generation, the young people listening, will see this happen. We'll see a different world. I think a more difficult world. It's not going to be a better world in terms of the environment. And there'll be more people. So, we need to do something different. And a big part of that solution is better plants. I mean, that's what I'm saying. It's not the only solution. Clearly, we need many innovations. But better plants is a big part of the solution, I think.
So you believe that awareness is also equally important because people are not taking it seriously?
Yes, well, I think, scientific colleagues, like I'm guessing many of the people listening, my scientific colleagues, we take it seriously. But it's the politicians and maybe the voters. And then in some countries, there's no democracy, so the voters don't have a say. But unfortunately, despite the disasters we see again and again with more powerful storms and droughts and floods and hunger and all these things, heat, and some countries have taken this seriously, but many have not taken it seriously enough. I think, well, you all know, you all can read the news, right? And there's these global targets and we're not reaching the targets. And we have the technologies to reduce impacts and to pull CO2 out of the earth with plants and so on. We have tools that we're not fully utilizing. And I'm hoping that as the situation gets worse, people will become more serious and we will, invest in this type of work, invest in young scientists, developing better agriculture, developing better tools. I'm hoping humanity will do that because it's a very grim future if we do not.
That's really nice. And then I would like to move next question. This is, what is your opinion on GM crops and do they have a future in attaining food security? Recently, we know that after 10 years of ban, Kenya approved the GM crops. So what is your opinion for that?
Well, I think, like I was just saying, Priyanka, we have a crisis. And so we should use every tool available to solve this crisis, right? The main thing is to feed those 800 million hungry people, to bring these people out of poverty. That's the main thing. And if GM is there and we can do that, great. Why not? Right? That's what I think. We should use everything. Now, that being said, there are some issues. Of course, there's not time to consider them all here today, but one of them is the GM is in many cases owned by corporations. And so there's a whole social and political and economic context of GM. One is that another is GM crops may displace the indigenous land races that have their own value. You see this in India, for example. You see this in Mexico. The Mexicans have all these wonderful maize land races, huge genetic diversity, being replaced by GM maize from America, from US. So that's a downside to GM, right? And we also have the fact that, as you can see from my own work, I mean, we have bred plants in Africa, Asia, Latin America without any GM because there's such huge genetic variation already. There's natural genetic variation. We don't need to improve crops by adding an alien gene. The genes are already there. And furthermore, most of the phenotypes of interest to me are quantitative. They involve multiple genes, although I've shown you examples where there's a single gene that's associated with the phenotype. Well, these things are always quantitative, right? So, so I think we have a nuanced approach. I think we should aggressively pursue any technology that helps us. But we should also be considerate of the native peoples, their food cultures, the economic issues associated with foreign corporations. And, I don't think GM is the magic bullet that some scientists think it is, because I'm talking about integrated phenotypes today, right? I'm talking about quantitative traits. I think the real challenge is not some magic new gene. The real challenge is thinking about a new rice, a new maize, a new pulse crop. It's going to look different to live in this future environment. What is that? How do we create that? How do we think about that? I believe that's the real challenge. In science, there's always these waves. So when I was a young graduate student, young postdoc, Priyanka, the big thing was genetic engineering and genes and genomics. That was the huge frontier. Well, that was already 35 or 40 years ago. So, and we've had huge impact. It's amazing what they have done. But it's no longer the frontier, I don't think. It's no longer the challenge, I don't feel. I think the challenge is the phenome. It's like the phenome is so complicated. I showed you Harini's work, 10 to the 16th phenotypes. I mean, come on, that's incredible. But that's what we have to deal with if we're going to develop new agriculture. So I encourage young people to think different, to be in the beginning of the next wave, which is the phenome wave. Sure, you should know genetics, you should know molecular biology, but you should also know soils. You should also know whole plants, because I think that's where the opportunities are.
But apart from the research perspective, if we see there is issues with the acceptance of GM crops.
Yes.
So what is your opinion for that?
Well, I think everyone has food sovereignty. I believe people should have a choice, and GM should be labeled. And, if you consult the people, maybe they want their traditional foods or they don't want GM food. You have to respect that. Food is more than just chemistry. Food is culture. People have strong feelings about food. Like we work in bean, it's very interesting because there are red beans and black beans and white beans and so on. Maybe the best yielding bean is something that people don't like to eat. Well, like one of our new bean varieties for Mozambique was black seeded. Well, in that region of the world, that has a bad connotation. They don't want to eat black beans, or maybe they don't have electricity. So at night they're eating their beans, they cannot see them, you see? So we have to think of, okay, we have to develop food that people actually want to eat, right? So that's our work has involved social scientists, and we think about these issues, the beans that women like, the beans that men like to take to the market. So it's a very complex space. And I believe here again, my recommendation to young people is be open-minded and work in interdisciplinary teams so you have these other perspectives, right? Like you may think this gene is going to be great, but maybe people don't want to eat that, or there's some other issue you're not aware of that's going to prevent you from having impact. And I think if we look at plant biology in my career, it's had much less impact than we hoped, right? Sure, we've had great impact, but at the beginning of the genetic engineering revolution, they've had all these great hopes and dreams, right? And yeah, in the US, we have Roundup Ready and so on, but what's happening in Africa? What's happening in Latin America? Has it really revolutionized agriculture? I don't think it's met our expectations, So we have to think of, what I'm saying is we have to think of food in a full context and not just as a collection of chemicals.
Perfect. So the next question is, what is your opinion on digital agriculture? Is it feasible to combine artificial intelligence and agriculture?
Absolutely. And some of the work I showed you today actually involves artificial intelligence and machine learning. And for example, Harini's optimization work and some of Ishan's work, when you consider these very large decision spaces, many phenotypes in many environments, even if you're working at the molecular level, many genes and pathways and transcription factors all interacting, it's beyond the human mind. It's beyond empirical research. We need powerful in silico tools. And here's another feeling I have about what young people should be thinking about. Please learn something about computational biology. You don't have to be a programmer, but you have to be familiar with these tools because they will be so important in the future to help us think about these complex problems. Like, how does this gene affect this phenotype? How does this phenotype integrate with other elements of the integrated organism? How does that interact with the soil? How does it interact with elevated CO2 and management? How does it interact with what women want to eat and cook for their families? I mean, this is a huge decision space. The computational tools will exist and are being developed, so we can think about that. We cannot just believe these computer outputs, as we see in my research, but then that guides your field research to the most productive areas. And I think that's going to become more important in the future, computational biology.
But what I have seen, plant breeders always believe that computational biology, or we are talking about the high throughput technology, which is nowadays are very popular and evaluating a lot of plots in a minutes, in a no time. But do you think that it should be like a 50-50%, or it should be a more towards the computational biologist, or we should reduce the plant breeding work as a physical what we do?
Well, I think that, Priyanka, I think that there's no substitute for the judgment of a human being, a breeder, who has an idiotype, who knows what she wants to achieve, who knows what that plant should look like. It's almost like art. You kind of know what you're looking for and that you go in that direction. And no computer can do that for you. You use the computer doesn't use you. And let me also say, you're talking about high throughput phenotyping. There's huge potential in high throughput phenotyping, but there's also a huge amount of nonsense, of things. People are measuring things that are not logical. So there's a lack of physiological understanding. There's a lack of organismic understanding. So you can do genetic analysis on anything, but does it really make sense? Is it a phene? That's one reason I use this old word phene, because that's an element of the phenotype. It's not 6 things together, it's one thing, right? And so I think we have to be more disciplined and more precise in the way we think about phenotypes. And I believe that this brute force selection or genomic selection on yield or all these massive high throughput approaches, they will fail, as they did at CIAT when I began my career. They looked at hundreds and hundreds of thousands of beans and they failed to find anything better than this one plant because they weren't thinking about the organism. They didn't have an idiotype. They weren't thinking about mechanisms. And so we come in and think, no, let's take a more organismic approach, a more physiological approach, and now we can make progress. And I think, that's it. And I don't blame breeders necessarily because they can't know everything. Usually they understand pathology and genetics, of course, but they usually do not understand stress physiology and so on. That's why we have to work in teams, especially in developing countries. We have to have teams of a breeder working with the physiologist, working with the biotic stress people, working, as a team, working with social scientists. We can't do this with just one person. And I feel like the best breeding is still, from the first Green Revolution to what we have done, it's still idiotype breeding where you have a concept of where you're trying to go. And that takes a person. That takes a breeder who knows what she wants to do. It cannot be replaced by some kind of algorithm, That's my feeling.
Actually, yeah, that is very good. Like, can root idiotype can be implemented in practice?
Well, that's what I'm trying to do in my career. I mean, I'm not a breeder professionally. I'm not in a company. So once again, everything we do is to try to solve a problem. We were doing a lot of work, writing a lot of papers about root phenotypes that were useful. None of the breeders were taking them. So we started doing our own breeding, right? So we show people, look, we can do this, see? Now, so I'm not professionally a breeder, but we have done breeding and it was successful, showing people, see, you can do this. So now next year I retire and hopefully some young person listening to this, okay, yes, Jonathan did it, so maybe we can do it. I mean, it's just like a pilot study, But you definitely can breed for better roots. And that's in the case of drought and low fertility soils, which limit most of the earth, that's definitely going to help. Breeders know this. When I began my career, are roots important? Of course, every breeder knows roots are important. They just don't know what to measure, or they don't know how it's going to affect performance. It's too hard. Their life is complicated enough, right? So we've tried to develop these simple tools and ideas. And, I think some of these things like long root hairs or root cortical parenchyma, it's like, okay, just use them. They're ready to use. They're not difficult. Just use them. You will have success.
Actually, that's what I faced like in person when I interact with some breeders. They said, oh, does it really possible to bring in a practice? It's like just identifying some QTLs for the root genes, or then how it is going to happen. So somehow we need to look at the deeper side. That's how we can bring into the practice. And I could see your fascinating work on roots. Like it's, I think your, how did you do that? I mean, it's really inspiring to bring into that level. So that's somewhere.
We have. You're right. I mean, and a lot of the root studies and root genetics over the many decades, they haven't had any impact. And I think there's different reasons for that. But that's one reason why our work in Mozambique, it's not the only example. I mean, people in the CG system have been looking at some root phenotypes, aluminum tolerance, and so on. But we wrote that story up. So there's a paper we wrote about what we did in Mozambique with our partners in Mozambique, looking at the social science, looking at the agroecology, looking at erosion and nitrogen fixation, looking at the root phenotypes. How do we breed this? And without fancy molecular tools. This is a simple breeding program, direct phenotypic selection, but it was a success. And I think this is a model that can be used anywhere by anybody. Like any breeder on earth can plant some seeds in a paper towel, and after a few days, look at the root hairs with their eyes or with the magnifying glass. And they can see, I have long and short. I want the long ones, okay? You don't need sophisticated genomic prediction models. You just, so I think, hopefully if there's one outcome from my work, in addition to the amazing young people I've worked with, if there's one outcome, is just to show people, yes, you can do this. It is not so difficult. If Jonathan can do it, you can certainly do it.
Perfect. So now I would like to move a second part of Q&A session, which is like a frequently asked questions from the next generation. So how should a scholar handle long period of experimental failures, unsuccessful events?
Experimental failures.
Yes.
Is that what you're asking? Oh, that's
Yes.
the best thing. The best thing is a failure, because if you've designed your experiment well, a failure can be that, we had experiment in Uganda once where a hippopotamus came out of Lake Victoria and ate our plots. So, okay. Actually, they ate some beans more than others. So we could have, if we replicated, we could write a paper about hippopotamus resistance. But in that case, okay, we lost the plot, we had to plant again. So there are sometimes you just have to do it again. But in terms of when your results do not meet your hypothesis or your expectation, that's the cool part. That's the new part, right? That's where you can find new things. That's led to much of our work. I mean, like I was trying to say, we didn't think 30 years ago all these things that I've showed you today. We just discovered them because, wait, we tried something. And, for example, once we saw Renkema was useful, we did experiments with the Renkema, and we thought, wait, the Renkema doesn't explain everything, so there must be something else. And then we realized, well, there's the number of cell files and there's the cell sizes, other elements of the cortex in addition to a Renkema. So those became new things, right? And so I think that when your so-called experimental failure means that your conceptual paradigm is inadequate and you need to expand it somehow. And that's the fun part of science, the creative part, right? Rather than just continuing to hammer the old ideas is when you can develop new ideas. So I think you should always open your mind and listen to your plants and look at your data and say, what is this? Maybe I was wrong, but now maybe I can have a better and new understanding by looking at these results.
So in that case, what is your advice if during that process, if students give up, like rather than like experimental failures, they give up, oh, this is like a research, and how to motivate themselves for that kind of work?
Well, it's one thing you notice, Priyanka, when you work with many, many young people, as I have, that, what are the qualities of a successful researcher? And maybe you think initially, they have to be very intelligent. I don't think that's the main qualification. Yes, okay, you have to have some intelligence. But the most important thing is discipline and persistence, okay? If you give me a brilliant student who's not persistent and not diligent versus average intelligent student who is persistent, I will take the average person every day, because science is like, okay, you have one moment of an idea and then you have 6 months of hard work. You just have to do it. You have to persist. You have to be disciplined. And, you are using scarce social resources to do science. If you don't want to do this, do something else. Let somebody else do it, right? Because there are not many scientists on earth. There's more problems than scientists. So those of us who are privileged to be in this role of doing science, you need commitment and dedication. If you don't have it, then you should be doing something else. I mean, that's for you to decide, right? But certainly you need to persist. You need to have faith. You need to have an open mind, and you need to question your data, but you need to have confidence that you're on the right path, right? Like I never doubted that my motivation to help people by developing better plants. I never doubted that. Maybe I would have failed. Maybe I would have not succeeded, but I would have kept with that focus, because that's my path, right? In India, you'd say that's my dharma, right? It's my path. And so if you're a scientist, you definitely have to have this feeling of, oh, I like this. This is my path. Otherwise, it's just so much work. Why do it, right?
Exactly. So that is like comes to next question. What qualities and concerns should we look out for when we when you initially when any student initially start their professional career or collaborations? So as you said, for persistence is very important more than intelligence. So what other qualities you look for?
Well, I think that a very important quality is creativity. You know, like I was saying a moment ago, there's a lot of science that's just like filling in the gaps or doing routine things, but on a new system or something. But then the really fun part and an important part of science is creativity, having a new idea, new approach, new perspective. And that's easier for some people than others to have this open mind, right? To be willing to go into new areas you don't understand. You know, I always feel like if I really understand what I'm doing, then it's not novel enough. I always have to feel like I'm in the edge of just not knowing what I'm doing. Then I know, okay, I'm on the forefront, I'm pushing myself, right? I go into new things because that's where the science leads us. Maybe that's not what I've done in the past, but you just have to do it. So this ability to have an open mind and to be creative is very, very important. Another element is just the diligence and the discipline and the persistence, because science takes a lot of dedication and hard work. And another element that is perhaps not emphasized enough is your ability to work with others, because successful science almost always involves collaboration with other people, other cultures, other ethnicities, other perspectives, men, women, older people, younger people, people from different disciplinary perspectives, right? And that's difficult. I mean, to be honest with you, the most challenging thing I do every day is working with other people. It's not the science, it's other people. How to motivate my own people, how to collaborate successfully with people somewhere else. I mean, this part, you know, in primates, we're primates, right? In primates, the size of your brain is related to the size of your group. So, you know, humans have a big brain because we deal with a lot of people, right? So this is important also. You need to learn how to communicate with people, to cooperate with people, to understand people. This is so important in science, in scientific success, right? So there's intelligence. You need to have some intelligence, but you need to have discipline. You need to have motivation. You need to have an open mind. You need to be creative, and you need to be able to collaborate and work with other people. I think those are the essential things.
I'm sure our next generation is listening that, and they are getting a lot of insights with that. So I would like to move to the last question. What would your words of advice be to the student and scholar who are new to the field of plant science research?
Well, I would say that I have a great deal of respect for you. This is a difficult path, but as I am saying, it is such an important path. I mean, it's so important for humanity to solve these problems. And in a sense, you are very privileged to be in a position where you can have such huge impact with your work. Your work can have such meaning. I come back to what the ancient Greek philosophers thought about what is happiness, what is a happy life, okay? And so their definition of happiness was the freedom to engage, to employ your talents in work you find meaningful. That's it. Their definition did not include success or recognition. It was freedom to do meaningful work, right? And so I think this is science. This to me is science. Nobody's, you know, telling me exactly what to do or how to do it. I'm doing it and it's because I want to do it, and it's meaningful because it can affect the lives of so many people. So I think really it's a very privileged position to be a scientist, a very important position. You can have a huge impact, but you have to take it very seriously. And like I said, I have a great deal of respect for this new generation because they will face huge challenges. And you have the potential to make a huge impact with your life's work. So I hope that everything that we have done in our work is just making a few little tools and ideas that maybe will be useful, but you will have to do so much more than what has gone before to solve these problems.
Thank you, Professor Lynch. And somewhere I would personally say that the researcher needs an engaging brain. It's not like 9-to-5 job, what they do, like what they see the other people are doing. They need an engaging brain every day when they are sleeping. going to sleep, they have to think about their experiment, what they can do, new things. So this is like this. The research is like research. You have to research all the time. So with this note, I would like to ask Shubho, is there any questions from?
Yes, but as you know, many students are listening this talk, and they are also replying instantly. So one asks you how many hours daily work he expects from a student. so that is the point. You need to love the work, not the hour. No one expects in our way. So yes, I know that type of questions can arise. And
Actually, I would like to add before Professor Lynch give the answer for that, because agriculture research people say they work in acres, not in hours.
Yes, and another question is that what we think that in developing country mainly, we are very obsessed with the technology, not the problem. As my point of view, because we are very fascinating now. One question is how I can enter the world of CRISPR-Cas9. So he is not thinking about the problem; he is thinking about the CRISPR-Cas technology. So it's a common nature in most of the developing country students, as I learned from Bioingene platform. So here is I wanted to ask this question to you: should we focus on the problem or in the technology? Because as your talk revealed that problem is the major, then you invent that technology that way. So these two questions from the students, I guess, and then I will ask two more questions from that.
Okay, yeah. So the first question is about hours. I don't expect anything from my people in terms of hours. I do not micromanage them. That is my style. I think science is a creative process, and everyone has their own way of doing science. And furthermore, it's very important. I think a real risk for young people in science is burnout and stress and working too much. And like Priyanka was saying, maybe you're thinking about it in your sleep, maybe you're dreaming about it. To me, that means you have to relax, because this is a creative activity. Your best science will be in a moment of creation and not just hard work where you can't sleep and you haven't taken a day off, you haven't seen your family. That's bad. My daughter is a painter, and there are weeks and maybe months she does not paint. I say, well, how, Lori, have you been painting? No, I just don't have any inspiration right now. And then she has inspiration and she's painting a lot. So, we don't scientists do not think of ourselves as artists. We think of ourselves like factory workers. Okay, I have to put in 60 hours a week. I think that's a problem. I mean, yes, sometimes science takes a lot of hours, but we also have to manage our creativity, our sense of well-being, the whole person, our families, right? I think the most successful people are people who have a whole balanced life and they're healthy and they're not just working all the time. And everybody's different. So I personally don't try to manage that at all. I just say, look, let's get this done. However you do it, it's up to you how you do that. Maybe it's many hours, maybe it's few hours, right? So there's a question about time, and I think, we can overdo it. That being said, most young scientists I know are working, 50 or 60 hours a week. But I think when you get more than that, I think it becomes counterproductive. It's not sustainable, right? And I don't think older scientists should demand younger people do that. I think that's abusive, So there's that. And then the second part is about, problem approach versus a technology approach. And I think, science is like an ecosystem. We need a diverse ecosystem. We need many kinds of scientists. We need people to develop new tools. We need people to use them. We need people to develop new concepts in one crop and then people to see how they work in other crops, right? We need a whole community of people who together are making progress. In my career, as you've seen in this brief presentation today, I've always taken a problem approach. I said, look, I'm trying to solve this problem. I'll do anything to solve that problem, including things that have not been done by anyone else. We invented our own things, invented our own ideas. That seems to work for me. but then other people are, experts in some particular approach. That's fine too. And maybe they're good collaborators for people like me. So like when we thought of the idea of using a laser, we went to an expert in lasers. All he does is work on lasers, and we, worked with that person to invent this. So I'm glad he was just an expert on lasers, but I'm glad I was not just an expert on lasers. I mean, so I think what I'm basically saying is we need all different kinds of people. We need all different kinds of people. But I do, just for the young people, I should emphasize, of course, now looking back over 40 years of science, like I can do, is that science is wonderful because it changes, it evolves, and the hot tools and approaches of today are not the hot tools and approaches of 20 years from now. So I advise you not to become too focused and attached to one method or one approach, because that may no longer be so useful in the future, right? So keep an open mind and keep learning things, so that you can still contribute even later in your career.
Okay. Sir, there is two questions. Repeatedly, I can see in that way. One is a relation to nutrition, because root is important in that way. So many people they are trying to ask you your opinion on root in monocot and dicot. And is there any relations between monocot, dicot in terms of nutrition and uptaking, or because overall I can understand from their questions is monocot, dicot root system and their nutritional uptake. Is there any difference, or what is your opinion on that?
So the question is about, are there differences between monocots and dicots in nutrition? Is that...
Yes, in terms of nutrition uptake.
Yes. In fact, there are differences within monocots and within dicots, and even within one species. And so that's what I was showing you, that, within bean or within maize, there are large differences, within rice. But certainly between monocots and dicots, there are large differences. And that relates to something I said a moment ago, is that we need people to work in many different crops. Shubha, you're working with jute. That's an important crop. We need to figure out jute. We cannot just all work in maize, right? And I think this is, in my career, there's been a huge fascination with these model organisms like Arabidopsis, especially, and that's been wonderful. But then if everyone does that, like in the US, you see like most of the plant biology community suddenly works on a model organism or two model organisms. Well, Arabidopsis doesn't have mycorrhizas, and it's a dicot, and it has a very simple root anatomy. It doesn't have a parenchyma, and everything is shallow in Arabidopsis. It's a tiny plant. So many of the things that we have researched, we would not have seen if we just stayed with the Arabidopsis, right? So the question was monocots, dicots. I mean, we have to think about the biology of all these plants. Now, all the work I was showing you have to do with annual herbaceous plants, crops, annual crops. But what about perennial crops? That's a whole different thing. The root biology of perennial crops is totally different than annual crops, right? So we need people to think about these things. I mean, we need all these people. And, that's what I'm saying. You look at the textbook, you see a picture of an Arabidopsis root, or you look at my papers, you see maize roots, but then you have to think about the biology of your plant and your soils and your environment. And that might be different, and there's things to discover there, and that's where the science is.
Yes. And one question is related to photosynthesis and root system, because people are mostly, as you said, that they are working on upper part of the plant. So they are wanted to know your opinion on cytochrome complex and root. Is there how they can relate, or is there any relations in that way? It's just opinion. They are not asking any.
Yes, there are relations. I mean, I've been saying you have to think of the integrated phenotype. That includes the whole plant, also its neighbors, right? In agriculture, we have fields of neighbors. We have 9 maize plants per square meter or something. So we have to think about that context. Photosynthesis is clearly important. We think about carbon costs and all these things, and we measure photosynthesis all the time. In my own case, I'm glad we have done work on photosynthesis over the years. I'm glad I have not continued that, because there are so many smart people working on photosynthesis. So what can I contribute? This little guy at Penn State, what's he going to do to add to all this huge knowledge on photosynthesis? So from that perspective, you have more impact with your own work if you work on something different, right? The same with, all this interest in molecular biology in my career. So we work on the phenome. And, that's more challenging to get your grant funded or your paper published, but you're more likely to do something new and different, right? So sure, photosynthesis is important. Do I think it can be improved for agriculture? I'm very doubtful. Because, like I said, even in rich countries, maize, the most important crop in the US, most richest country on earth, maize yields are only half of yield potential. So we're not using the photosynthetic potential we already have. Why is it half of yield potential? Well, there's water stress, for example. Okay, so now it's no longer photosynthesis per se, we're looking at water, you know what I'm saying? So I think this issue of stress is how we should view photosynthesis. I mean, there's room for upstream basic research or just understand plants regardless of agriculture. Sure, fine. But if we're trying to improve agriculture, we have to think about actual problems that actually occur in agriculture. And in that case, I am not aware of a single agricultural system on Earth that's limited by photosynthesis per se, that's limited by photosynthetic capacity, because they don't have enough water, it's too hot, there's not enough nitrogen. These are the limitations, right? So I think, in a world where there's plenty of food, we have the luxury to research whatever we find interesting. But in a research where there's so many problems and there's so few agricultural scientists, we really should be more disciplined and say, okay, I should work on something that's going to be important. Maybe it's a disease or a nematode or who knows, but it's something that is relevant to field problems, I think. And in that process, we'll not only have impact, but we will also discover new science, you see. That's my approach anyway.
Oh, okay. That's it. Yes.
Okay, that's the end. But I would like to read out one comment, very interesting comments we have received on YouTube. One student is saying, I wish I had you as my guide. My guide only does micromanagement. He's more interested in hours of work you do per day than real science.
Well, I'm sorry. That's the part I said about dealing with people can be difficult. But someday, hopefully, you'll be your own. You don't have this mentor anymore, and then you can do it your way.
Yes, but sometime there are a lot of such comment and quite interesting. But I wanted to add something that when you will be guide, like now I'm feeling these things. When I am a PhD student, sometime I thought that why my sir give me the project just 2 days before the submission of date? Why DBT not asking it for 1 month? Then I can make a good project. But now I realize that DBT actually asking 2 days before the 2 days. So now I am feeling that, okay, funding agency asking just 2 or 7 days before that requirement. So definitely that time I'm thinking that maybe my supervisor forget to send me, and now he's sending me to me for that make the project in a 1 year project in the 2 days. But now I can understand that it happening that way. So obviously we should not encourage in that way, but it's
I guess proper communication is very important with
Yes.
your supervisor in that case. So
Yes,
and-
I think, yeah, and you can have an abusive advisor, but I think in general, once again, have some perspective. I mean, I feel very privileged to have been working in science, partly because scientists are general wonderful people. Most scientists are committed. You don't go into science to be rich and famous and powerful and wealthy. That's a bad decision, right? I mean, you want to do those things, you go into business or, politics or something, right? So I think scientists are good people generally. I like scientists generally, right? So, they have good intentions, even if maybe they're difficult people, they have good intentions. So I think we have to be charitable and appreciative, Sure, there are difficult people, but it's probably worse in the world of business, is what I'm saying, right?
Yes, and now you can see, Priyanka, the effect of Sir's talk that can I have you my supervisor in PhD?
I am retiring next year, so I'm no longer taking students.
Yes. So they are very helpful. Lots of people are acknowledge your guidance because they find it's very helpful. Specifically, the freedom to do meaningful work in peace. Absolutely, they are loved it. Your talk in this comment, and we're very happy to bring you in that such 50th webinar, and it's so interactive. Before the starting, we talked to Professor Johnson that it will take nearly 2 hours. That time I'm thinking, is it more or less? Now you can see that time flies. We almost completed 2 hours talk in that way. So it's lovely. Your and lots of people are comment on your imaging, because we are not generally seeing such type of imaging, and specific, and it's sometimes we overlook that type of journal because maybe we are not doing that work. But this platform is give us that type of freedom that we can learn such thing, and I'm really admire, and I'm sure lifelong your imaging and image technique for root will stay with me. It's a great inventions that you are trying to do in that system, because there are a lot of questions. So when we are not visible anything, their questions is more like root. It's not visible in upside, and lot of questions we have in that way. But it need techniques and obviously need the instrumentation in that way. Lot of appreciation for your talks, and I'm collecting all the questions, and people are really loved your opinion on various aspects, and all are loved your sessions, and these comments are live. I'm thinking more than 300 people are lively watch this webinar, and it can be viewed as it's available on our YouTube channel. Thanks, Priyanka, for bringing Professor Jonathan in this platform. Otherwise, maybe I'm not able to go for that. Say, thanks, Shoma, thanks, Priyanka, and thanks, Jonathan, for give such a wonderful lecture, such a innovative and informative. It will give, and when I see your presentation, style of presentation, presenting, and acknowledgement. Every slide has a picture of a JRF or SRF. It's a great. Means they also loved the work. They also happy with their work, and I learned it from you. I will try those. I have a 1 JRF right now, but I'll do that. I'll do that in my career. When I'm presenting something, I'll put that picture in that way. Thanks, Priyanka. Can you? Yes.
Yeah, I would like to add, certainly it was a great webinar. And it's like not only as a scientific, definitely scientifically it's very advanced, but as a person, as a like how one should be a mentor, and what are the characters they should develop when they go into their research. That is very wonderful. So I really appreciate for accepting this invitation. Thank you so much, Professor Lynch.
Thank you for this opportunity. I'm honored to help serve these young people whose work is so important. So I wish you all success with your work. Thank you.
Thank you so much. Thank you.
Thank you.
It was wonderful. Thank you.
Thanks. Shoma, you may close the session and keep the recording. Thanks, Jonathan. Namaste.